So let's sum up. What have we seen so far? We've seen 4 complexes where 3 of them are basically pumping protons through the membrane, and that is the key part. That and then the oxidation of NADH and FADH2 towards the end of this. So this leaves us with a huge membrane potential that we will utilize. Does this occur other places that I just said in the beginning? Yes, it's also occurring in bacteria. It's slightly different. And as you can see here, the cytochromes are then cytochrome O and cytochrome D. So there's slightly different cytochromes, but the principle is the same. We have NADH, FAD cycle here. We have ubiquinone and ubiquinol cycles. And we have protons that are pumped out into the periplasmatic space, which is like this intermembrane space in bacteria, so that we create a membrane potential over a plasma membrane. Membrane potentials. Membrane potentials is key. Membrane potential is vital and is one of the ways that we create chemical energy. So here in this figure, you see the ATPases from bacteria and from, in this case, yeast. And I put up a link to a YouTube video on how these actually function, so how these rotate around themselves. It would be in this direction. So the key part here is that we have the F0 part, which is the proton channel and the membrane anchor, and the F1 part, which is the catalytical domain of the ADP to ATP. So splitting it up in the more details, the key part is the head piece part here, which is an alpha beta domains that are the one that are initiating the ADP to ATP. So these do actually not rotate. These just open and close, while the rotating unit here pushes the different alpha and beta subunits to different conformations. So that's what we've talked about before, that we have some kind of electron or proton or some kind of ionized change in our enzymes that changes conformation of one part of the enzymes, which pushes on to the other part of the enzyme down here. And then we have the stator here, which is kind of like holding everything in place and is immobile. So here we see what I talked about just before, that we have the rotor here turning around inside the alpha and beta domains of the headpiece, and the turn induces conformational changes of the various parts, and this conformational change then opens or closes the active site. So if we look at B1 here, beta 1 here, the active site is open, ATP can be released, and ATP and phosphate can enter after that. Then we have the form a closed environment called L, where this will close up gradually here, and in the end that will push the two sides kind of like together over this and push the ADP and the phosphate closer together to form the ATP synthase. So so that's the way it's going on, and so that is what is trying to be depicted here, that upon the release you get this L formation here, and it's getting closer, and that then goes around, and then you get the release down here that we had before with the T, and then we get the release here. So that's opened, closed, and a transformation status here. So the gamma subunit, the rotor, goes counterclockwise, which is normal for all of these ATP or the proton-driven pumps in this situation. Another way, so how is it then driven? What is the way to show how is it driven? So what you have to imagine is you have this membranes part here. We had this part, this purple part is part of the stator. That's the proton comes in inside this the positive charge. So inside this channel, there's another positive charge which induces that the proton turns to the pushes the whole molecule in its presence to the right, no, to the left here. Sorry. Um, and that because you know two pluses don't really like to be close to each other, so one will go the one way and the other will go the opposite way. So the protons will then travel around in their holder until they are falling out down here into the matrix again. So they will come from the intermembrane space or the and then put into the matrix slash cytosol depending on its eukaryotic or bacteria. That's how it is formed in here. And yeah, so this is just basically the whole principle behind it and what leads to the rotation of the ATP. Now, the pH in mitochondria, of course, is very much driving this force. And here's just shown how this can be done. Basically, what we really want to have is the pH in this area, so the intermembrane space, which gets around down 5.8 to 6, in order to have enough membrane potential to drive this whole process. So if you see the cytoplasm, if that is around 7.3, if that is the same in the matrix, then it will be 7.3 all the way through, and we will not have any membrane potential. So we really want to keep this as a lower pH than both in the cytoplasm and in the matrix. And that this is where these loops in the inner membrane actually help us, because these loops creates an area where we pump out the protons into this loop, and that up concentrates the protons in this area only and not in this longitudinal area out here. So only inside the loop. And then in the end of the loop, we have a high concentration of ATPases that kind of like pumps back the protons into the matrix. So in the ATP ACE, we have this reaction. We have ADP as a substrate, we have free phosphate, and they need to come close together to form this pentacovalent intermediate, as we see here. So here we have actually the pentacovalent phosphate group here. That is quite unstable. So unless they are kept close together and water is not present, so if water is present, they will just be hydrolyzed again and we'll go back to the ADP phosphate part. But if water is not present, this is where it's really important that the enzyme keeps the rest of it out. We get ATP formed and we get water released from this reaction. So this part is reversible, of course, but it is also reversible the other way around. And these are the steps inside the ATPase, which is ongoing all the time when it's turning. Now, as I said before, it's really important that this is regulated by this membrane potential. So one way that this is regulated is actually by phosphate translocation. So obviously, if we don't have phosphate present down inside the matrix, we cannot form ATP from ADP. So one way is that there's the membrane potential also helps in the phosphotranslocation pump to pump phosphate through. So the protons is a co-carrier of phosphate through the membrane. So that's one way to get more phosphate inside. But again, this requires that we have a membrane potential because we have to have more protons on the outside than on the inside. That is one way. Then there is the ADP ATP translocase. So this pump at a high proton level is transporting ADP through and only ADP, but at a low proton level, it will not pump the ADP through. But again, as a high proton level on the outside, it can also pump ATP on the outwards. So one way that this has been studied is the use of phosphoric acid. So phosphoric acid is a phosphate group, and then you can transport the phosphoric acid together with proton through the membrane, and that way you actually create an even larger membrane potential. But that was studied using this. The What was discovered was there's an antiport function in this, and that's actually the transport of hydroxyl ions. So to some extent that makes sense, but that will actually lower the pH out here, because then we'll suddenly have hydroxyl ions that will go together with protons and form water, and we have less free protons for the membrane potential. So that's another way that this can be regulated. But the main regulator is basically this with the phosphate. And so with the high levels of protons, we will have a lot of phosphate pumped through. That enables us to make a lot of ATP in the ATPase. We, in the electron transport, also talked about the NADH, and maybe you remember that some of the NADH during the glycolysis is actually made inside the cytosol. Most of the NADH, all the NADH that are produced in the citric acid cycle is made inside the mitochondrial matrix. So that's not an issue for complex 1, but all the NADH that is made in uh, the glycolysis uh, through the is needs to either be oxidized in a different form or recycled in another way. So the eukaryotic way to do this is that you have a dihydroxyacetone phosphate, again a molecule that we know from glycolysis. We have the dehydrogenase, which utilizes NADH and NAD, or utilizes NADH, so it's oxidizing NADH into NAD. And the reduction of into the glycerol 3 phosphate enables that the the glycerol 3 phosphate pump, can pump this into the intermembrane space. In the intermembrane space, we have a mitochondrial glycerol dehydrogenase. We have another glycerol dehydrogenase, which is not sitting in the mitochondria, but sitting in the cytosol. But the mitochondrial glycerol dehydrogenase will then transport the or change the glycerol into dihydroxyacetone phosphate again. And that process um, requires FAD. So that reduces FAD to FADH2, which can then be oxidized using the ubiquinol cycle. This is the reason why in eukaryotes we actually have two less ATP production, because the NADH from the formed in step 6 in the glycolysis is in the cytosol, and so there the reducing any elements needs to be transported into the intermembrane space and into the mitochondrial matrix, and that costs because the NAD is changed into FAD, and FAD, as you might recall, is an element of complex 2 and loses at least 4 protons in complex 1 that we otherwise could have transported. So that is the main change between eukaryotes and prokaryotes, because in prokaryotes you of course don't need this, because there everything takes place in cytosol and you don't need this transport over the membrane. So this is how that eukaryotes usually is depicted as having 30 ATP outcome and prokaryotes 32, because of this loss of 1 ATP per NADH from the second part of the glycolysis. Another shuttle that is also well described of NAD, but not so much used, is this malate aspartate oxaloacetate shuttle. This occurs in the liver and in the heart. And why has mammalian organisms come up with a secondary way to cycle the NADH-NAD? Well, of course, in the liver and the heart, if you think about it, these are high energy requiring organs in our body, and thus, if we could come up with a way that instead of that we just saw, where you lose actually 1 ATP per NADH, but then just do the cycle of here to the NAD, H to the NAD, transport malate across, and then take malate back to oxaloacetate and into aspartate, but then we use the NAD plus in the mitochondrial matrix to create NADH. Well, then we kind of like don't lose anything because we just get NADH out of it again. There's of course a cost here, and that's a transport of malate and the antiport here of aspartate glutamate transport. So we have to have glutamate production from somewhere else in the body, and that's why this is only in key vital organs, and then we rely on glutamate and alpha-ketoglutarate production in other parts of the body. So there is of course a payoff here. It's not a full cycle, even though it might look like it, but it do cost something. As a whole, it's a smart way to save 2 ATP in vital organs. So this is the table from your book where it shows where do the different ATPs come from. And I just want to show that these two down here, so we have the liver cells where we get this oxidation of 2-NAD, and the muscle cells where we only get 3 of them, right? Because we kind of like have the NAD FAD cycle and not the malate aspartate cycle. These are of course not present in prokaryotes, and in most eukaryotes these are also not present. So these are normally most eukaryotes just have the glycerol 6-phosphate shuttle active, and very few use the malate aspartate shuttle. So there's a difference here between the different cells and eukaryote cells. But prokaryotes don't need this, so they basically just have these 5 in total instead of having this loss of 2. Yeah. So we are done. We've been through all the way from glucose to water. We have glucose fully oxidized into CO2 and water. We have the oxidation of oxygen. And as you can see on this slide, there's a lot of regulatory elements. We already talked about those in many places. The main story here is that ADP, sign of low energy levels, induces the whole pathway. ATP is the contrary, it reduces the pathway. So with that concludes our discussion on or presentations of glucose to CO2. And we today just talked about the electron transport chain, NADH oxidation, FADH2 oxidation, ubiquinone reduction, cytochrome C reductions, and so forth. Transport of protons across the membranes. In total, we get a transport of 10 protons. And that leaves us with a potential to produce about 3 ATP molecules from one NADH molecule. And then for the FADs, H2s, we usually say that we can produce 1.5, and the NADH usually 2.5 per molecule. There's a bit of a loss because, as I showed before, we also need to transport phosphor across, and there are other elements that are influencing the actual production levels. So it's not just a straightforward NADH is 2.5. It's an estimate of the average between very many different cells. So just a resume slide if you're in doubt. Glucose being the key here, glycolysis, pyruvate oxidation into acetyl-CoA. There's these extra chance of oxaloacetate, glutamate, alanine. We can get something produced here. The citric acid cycle, which is not fully circular, but it could be made circular. So these are, look at all the arrows, not just the full ones. These are different fermentation steps. And finally, the oxidative phosphorylation down here. And basically, in biotechnology, you really want to cut away most of what's going on here and utilize pyruvate directly into your product. But that we will hear about in some other talks at a later point.